Dr. Patel is an assistant professor of biomedical informatics, Harvard Medical School in the US. His long-term research goal is to address problems in human health and disease by developing computational and bioinformatics methods to obtain reproducible high-throughput data streams spanning molecules to populations. Dr. Patel will tell us about this important novel approach. So please.
Great. Thanks so much for the invite here. I shared shared the stage with two spectacular speakers, and I'll try to at least keep you up to these last few minutes. It's an honor to be here. Thank you very much for the invitation to the Congress and to the Ministry of Environment here in Japan. So what I'll try to articulate today are the data analytic challenges that are going to emerge when collecting large amounts of high-throughput biological data that in our observational studies. And so what I'll try to base some of my arguments and describe the challenges on the years that we've studied genomics and genomic science, and how these tools can be applied to looking at environmental exposures and their interactions with genetics to look for new risk factors in disease and health. So as you know, as I try to tell our Genetics 101 students, that the name of the game, the reason for the season, is phenotypes are a function of genotypes and environment. So things from like disease, how genes are turned on and off, are a function of things that come down from mom and dad, or millions or so variants that we inherit, plus of course environmental exposures, infectious agents, diet, nutrients, all the things that we've talked about at length here today. But the thing is, we're really great at genomic investigation today. So as of this very old, outdated slide, 3,000 publications so far, looking at a data-driven investigation, essentially a search engine, agnostically, to search for variants, genetic variants associated with phenotype. And so the community has published over 40,000, or almost 40,000 of these connections between individual variants along the genome with phenotypes. And this is really has been accelerated due to cheapening technologies, more accessible genomic technologies to measure things at high scale. And I'd like to argue that there's nothing like this to elucidate environmental influence at the same scale, but a similar paradigm of discovery should exist. So again, back to Genetics 101, so why should a similar paradigm exist? So variation in the population of phenotypes is equal to variation of genetics plus environment plus their interaction. And we have this great indicator in human genetics, or in genetic population genetics, of this notion called the heritability, which is essentially the amount of variation in the population in phenotype that is due to genetics. It ranges from 0 to 1. So a trait that's completely heritable is something that's completely, almost genetically, totally genetically controlled. And those that are not heritable are those that are almost environmentally controlled. And again, the things that we care about today are things that have low heritability. So here I pulled down some heritability estimates from SNPpedia.com. They range from 0 to 100. So here on the bottom, you'll have things that are highly heritable, like height, type 1 diabetes, eye color. These are things that you can intuit that are strongly genetically controlled. But other things up here in this top area of the figure have heritabilities less than 50%, include things like diabetes, heart disease, and things that have a wide range indicating some interaction with the environment are things like autism. So this, I think, I hope to convince you, there's this large amount of phenotypic variation that we have yet to explain in the population with just looking at a handful of environmental factors at a time. And I think this provides a great substrate for discovery of new environmentals associated with phenotype. So let me tell you a little story about how we got there with genomics. So in 2001, we were able to sequence the entirety of the genome. 2001 to the current day, characterize how people are different along the genome, and then again have these new accessible tools for assessing how we're different, how you and I might differ along the genome. So for less than $100 today, you can get 1 million or so variants, almost 10 million or so variants measured along the human genome. This price will probably be 0 in the next couple of years, okay? And so now we have these, in just 7 years' time, you have this essentially this nice Google search engine, if you will, to look for variants associated with phenotypes. And so this is one of the first notable papers in Nature in 2008, looking at 7 diseases in a case control design of 14,000 people, and now we're looking at hundreds of thousands of people at scale, all right, now in just about almost 17 or 18 years after the sequencing of the genome. Very quick to get to those discoveries. But as the heritability estimates I just pointed out to you, they make a very little difference in clinical decision making. So how does this work? And I think I'd like to explain how this works, because it's a great example of how data science can exist in the new paradigm of exposome science, if you will. So this is a prototypical plot that you'll see from a genome-wide association study. This is from, again, that Nature paper published in 2008. That's a typo there. So here you're cruising along all the variants along the genome, 10 million or so. And here you're plotting the association between a variant along the genome in case or control status. And here you're looking at the negative log 10 p-value. So this is the lower this p-value, the greater the difference between your cases and controls in allele frequency. And you'll see these positions light up like the Manhattan skyline to denote association between that particular locus and your phenotype of interest. Okay, so this is a robust agnostic tool to use to look for, discover new things connected with your disease of interest with respect to genetics. And why do we like this in genome science so much? I think for all the reasons why I think data science should be a tool that we could use for environmental research. First of all, it's comprehensive and transparent. I'm going to tell you everything that I'm associating with my phenotype of interest in a comprehensive and transparent way that's transparent to the readership. It's multiplicity control for type 1 error. That is, we have this common agreed upon notion of what's signal versus noise in our data set. And of course, we open ourselves up to new things. So prior to the genome-wide association study literature, the candidate gene hypothesis era, where we pick your favorite gene of interest, maybe it's a gene along this axis here or a gene along this axis, has been proven to be almost all false. And it took a paradigm like this to really clean up that literature to find new robust associations with phenotypes of interest. But again, these new loci have very little clinical import. And so what I like to talk to you today about how we could use some of these tools and methodologies in environmental science, and I summarize some of the work that we have here today. So these are some of the papers if you're interested in looking at describing some of these perspectives that we have. So first I'll go on to for the promises. The promises are pretty obvious in that, you know, when you're doing high throughput research, you're opening yourself up for discovery. That's systematic. We hope that's reproducible, that you're replicating in different cohorts. You're controlling for that type 1 error or looking at for signal to noise ratio, but prioritizing for further study, hopefully in factors that you can in an experimental setting, like a clinical trial or a genomic study to look at model systems perhaps. So for example, let me show you some examples of this exposome-driven machinery. So here, in some of these examples, we'll show things from the National Health and Nutrition Examination Survey. It's this important effort undertaken by the Centers for Disease Control and Prevention, where in the United States they send these trucks like you might be doing with your GEX cohort at different places around the U.S., collecting sample in a cross-sectional fashion and measuring things like different indicators of environmental biomarkers of exposure and other clinical traits like serum glucose, lipids, and body mass index, and also linking them to some longitudinal outcome like cause of death. Okay, so what we did was in our initial studies is we took the 200 or 250 or so environmental indicators measured in biomarkers in the NHANES and associated with them with different clinical phenotypes. So for example, serum nutrients and vitamins levels, infectious agents levels taken from urine and serum, plastics and consumables, pesticides and other pollutants like cadmium and lead, and other quantitative indicators, and connected them with phenotypes all systematically. So in these two examples, let me show you things that kind of make this paradigm of discovery a little bit more obvious. Here we looked at all-cause mortalities as a first example. We took this NHANES survey, we split it up into two, one for discovery and one for replication. So for example, the 99-2001 we used for discovery, and then we used a Cox proportional hazards to look for time to death. And we use this false discovery rate, which I would argue is probably one of the most impactful pieces of biostatistical tools that we use in bioinformatics to ascertain signal from noise. And I'll show some papers on that in a few seconds for those of you who are collecting references. And then we take those that we found that at a low false discovery rate and try to replicate them in a left-out cohort for replication studies. Again, what is the false discovery rate? get into that in a second there. And we also try to estimate the variance, explain to the population or how much of variance in death or your phenotype of interest that you could find correlated with all your all your exposure values systematically. So again, the false discovery rate, I put this here for a reference for you. I think it's one of the most important papers for us in biostatistics and informatics. It's a way of estimating the rate of false discoveries at a given p-value threshold. So for example, if you say I have a p-value threshold of 0.05, and you have about 100 results you're getting at that threshold, how many of those results that I'm picking out in that large database of associations you might consider to be false discoveries? So this is from Benjamini and Hochberg. Already it has over 10,000 citations, and something that one really needs to think about implementing when doing discovery research. If you want more tips and about how this type of stuff works under the hood, I recommend this great review article from Bill Noble in Nature Biotech. So let me show you how this might look like in a EWAS type of setting or exposome-wide association study. Here we looked at 250 or so exposures, 246 actually, 243 associated with time to death in this Cox proportional hazards model. And so some of the prototypical things you might see are this false discovery rate threshold here. This is a signal to noise ratio, if you will. On the x-axis it denotes the risk for death. So here's the null, here your risk, the adverse risk for death, here's protection from death. And what I'll show are things that are, for example, replicated. So things that are in these black marks are, I'm sorry, this is dying here. The black marks are those that are replicated, that is found at low false discovery, but also replicated in the independent cohort. And the red are the very important sociodemographic factors I'll talk a little bit more about in a few slides here. And so if you just look at the picture of the National Health and Nutrition Examination Survey with respect to death, you see that socioeconomics play a huge role, as well as, of course, aging, or chronological age itself. So for a 10-year increase in age, you have, as you'd expect, a twofold greater risk for death, as well as individuals from the lower income quintiles are also increased risk for death that are almost equivalent to aging, chronological age of an increase of 10 years. And men versus females are also at about a 60 to twofold greater risk for death versus females, a very well-known biological phenomena. But then when you start to overlay some of these factors that we tried to derive in a data-driven fashion, you have indicators of nutrition. So for a 1 standard deviation increase, you have about a 20% decreased risk of death. Smoking kind of predominates the U.S. population with respect to death. So past smoking, current smoking, anyone smoking at home, as well as things that are predominant in the smoking matrix, if you will. So serum and urine cadmium, about 20 to 60% increased risk for death. And when I say the smoking matrix, I know this is also an elusive idea. I'll talk more about this complex correlation web of exposures in a few seconds here. But also things that you'd also posit before, like physical activity, one of the strongest factors connected with a decreased risk for death here in the NHANES. But then if you put all of these things systematically, all these 250 things into a model and try to explain the amount of the death in the U.S. population, you only get an R-squared of about 2%. So if you think back to those heritability estimates I just showed you, you have about less than 50% potentially explained by genetics, probably not even that much with respect to aging, and only 2% with some of these strong factors here. So a long way to go to explain death, even modulo random events in the U.S. population. You can do the same trick with other phenotypes. So here's another indicator of aging, telomere length. Telomeres are at the end of DNA that actually helps or protects it from replication, just also due to chronological age. And so similar idea here, you have a false discovery rate threshold and you attempt to find factors connected with disease. And here what I want to show you is that we find many things that were also found in all-cause mortality, as you might expect, because telomere length is a phenotype of aging, like cadmium, like indicators of physical activity, like VO2 max and pulse rate, but also new things like PCBs, for example. We wouldn't have hypothesized before that, in fact, longer telomeres are indicating of that are potentially good for aging, if you will, also connected with longer, with greater exposure to exposure levels with PCBs. So something that it could be discovered through this process that really needs to be evaluated either in model systems or in other causal settings like Mendelian randomization, which I'll hum a few bars in a few slides here. But these effects are also pretty modest. So you have about, for a standard deviation increase, you have a increase of about 2 to 8 years in the age, even after adjusting for age and age squared. And you're only explaining about a percent of the telomere length in the population by putting all of these 450 factors here systematically into a model. So a long way to go to explain telomere length and other aging indicators in the US population. We have more examples if you'd like to take a look. Of course, I share with you a link here. So briefly, just to summarize this first part, that I think it's possible to capture exposures in a high throughput way with new technologies, such as mass spec, which I'll talk also about in a few seconds here, as we move away from looking at a handful of things at a time, candidates, to being more holistic and comprehensive. And the name of the game is to try and describe more variance in the population, of which even with just almost 400, we're doing a modest job. So let me give you a snapshot of some of the challenges, one of which, of course, is we lack a unified high-throughput assays to measure exposures in a high-throughput fashion, like we do with the genome. And so let me talk a little bit about that. And I'll spend the bulk of the challenges section here talking about some of the biases that we might incur looking at large data sets that are time dependent, as we expect the exposures are, of course. So it is a challenge to measure exposures at scale at the moment, okay? And this is primarily due to heterogeneity in a large dynamic range of some of these exposures that we'd like to measure. So here's a great paper by Steve Rappaport at Berkeley, published in EHP in 2015, trying to assess the serum and blood exposome. And what he's trying to depict here, Steve Rappaport and David Wishart and colleagues, is the range of exposures that we need to measure is quite large. So it's over multiple folds in concentration, for example. And those that are of concern, for example, the pollutant factors, or of concern is everything, obviously, but some of these that we have very little data on, as opposed to others, are measured at very, very low concentration, or seen at very low concentration. And the others, more endogenous indicators, as well as other exogenous indicators of things like diet, occur at a larger concentration. And so the new paradigm is to use a targeted approach to look in mass spectrometry to look at some of these things at lower levels, and in a targeted mass spectrometry approach, borrowing from the tools of metabolomics to look at things that are of high concentration. But it is still difficult to scale this up, because the issue becomes when you're looking at it in an untargeted fashion, where it is possible to look at things at a higher concentration and multiple things, almost thousands of things simultaneously. It becomes a challenge to identify what the heck you actually have in your serum or blood. So these are things that need to improve with using these technologies to get to the level of the genome, for example. Other things include the big bias that we incur when looking at larger data sets. So things that we've seen so far are indicative of fragmented and small associations, which may also indicate other things like residual confounding and that kind of make the challenge to make causal inference. Here's a great example of fragmentation that we could see at large scale. I'll show you an example of that in a second here. Here's one from John Ioannidis and John Schoenfeld that they pulled from the literature. So here they looked at 50 random ingredients from the Boston Cooking School cookbook, and they asked, in observational studies, have been associated with cancer? And of the 50 that they looked at, 80% were actually studied in cancer risk, so you have some coverage there. But they found that there's weak statistical evidence, non-replicated, inconsistent effects, and non-standardized across all these studies. So this is pretty well articulated by this plot here, where they're looking at relative risk from different studies of these different types of ingredients pulled from the Boston Cooking School cookbook, of risk estimates that float all over the null here. And so it's really difficult to assess whether dietary factors in these observational studies, when using these non-standardized and non-replicated associations, are in fact in fact, something that we can intervene on and indicated of this fragmentation. And this can be amplified at large scale. So here we asked, again, with Johnny Ioannidis and our colleagues, Christina and Jan Sundquist in Sweden, are all the drugs we take associated with cancer? And so what we did was associated 500 drugs prescribed in the entire population of Sweden. We had almost 10 million people with time to cancer. And we looked at different modeling techniques with respect to cancer risk. Okay, and so here is our Manhattan plot, if you will, of drugs. Here are all the drugs that are organized in different classes associated with time to cancer. Of the 500 or so that we queried, almost over a quarter of them were correlated with future risk for cancer. If you ask me, do you believe that result? No way we believe that result, right? So a lot of these associations that we're finding in this ultra-powered large data set, looking at all the drugs prescribed in Sweden, could be, in fact, false positives, even though they have strong statistical associations. So it's modest also to point out the fragmented literature, is that there's a modest concordance between methods that one might use. So if you choose a Cox versus a case crossover approach, you're only getting 12 out of the 141 that agree when using these two approaches. So again, harmonizing methods and to make sure that we have a systematized approach for looking at these factors at larger scale. And most of these correlations, which I don't show here, these are p-values, but I'll tell you that they had low hazard ratios. So they're small, which may be indicative in this population of a large amount of residual and residual confounding and bias by indication. The other things include this dense correlational web that exists between exposure. So here again, we took the National Health Nutrition Examination Survey. We took 575 factors and we correlated one of them with one to another using a very simple Spearman approach. And then we organized all these factors in a globe to depict some of these correlations. So here, as an example, I show cotinine, serum cotinine, and its correlations depicted in the red lines with other factors that are also ascertained in the National Health and Nutrition Examination Survey. So this gives you a bird's eye view that higher levels of cotinine are correlated with higher levels of other volatile compounds, other hydrocarbons, and also some heavy metals as well. But if you start looking at the entire database of potential correlations, you get this large spaghetti ball, or this large spaghetti nonsense of correlations that exist in the data in this population. So you have strong correlations between hydrocarbons and phthalates, for example, amongst PCBs themselves, and it becomes a huge challenge to decipher of what correlations that you found are truly connected with your phenotype of interest. There, of course, these codependencies are strongly connected with things like shared environment, so individuals who live in the same household, for example, income levels. So we have a paper showing how these codependency globes change with respect to income, with respect to, of course, sex, and shared environment, as a postdoc of mine, Jake Chung, just showed, and is now up on bioRxiv as of yesterday. So you could take a look at some of these correlation globes, if you will, if you'd like, on this website here. I think, but amassing a database also enables us to understand whether your correlation matters for future study. So here, what I depict here is, we can ask the simple question: does your correlation or your hypothesis actually matter in the entire space of correlations that could possibly exist in your data set? So for example, here I show in this cartoon here, maybe P levels of phenotypes, maybe different types of diseases or clinical indicators, and different exposure factors here on the top axis here. And there are a lot of hypotheses that you could potentially explore in this database, if you will. So what this square is depicting. So it becomes a challenge of which ones do you test. Do you test them all? You test the ones in blue, maybe some that you had a prior hypotheses on. And what it becomes is this challenge of understanding how many possibilities or what the signal is from the noise, given this entire space of correlations you've essentially created in your large exposome cohort database. So let me show you an example of this. So here, a collaboration also with Johnny Ioannidis and Dennis Beer and Hugo Aschard, we looked at all phenotypes versus all exposures in the National Health and Nutrition Examination Survey to get a bird's eye view glimpse of what could happen in this scenario. So here we wanted to draw a map, if you will, of different phenotypes here on the long axis versus all the exposures here. And so if we see a correlation, we color it one color, and if we see a correlation in the opposite direction, we color it yet another color. And so what we look at are about 500 or so exposure factors versus 209 clinical trait phenotypes. And we looked at over almost 83,000 correlations, if you will. And so we also tried to sift through those that were significant using this false discovery rate approach. And we plotted all the connections, all these associations in this volcano plot here. So similar to what I showed before, here's a percent change for one standard deviation in exposure. And you get, of the 83,000 that we queried, about 12,000 that were significant by this threshold here, so 6% in yellow. And so you get this on average. If you were just selecting a correlation from your database, you're going to get on about 6% of them are going to be significant at this FDR threshold. And 95% of the associations exist between -6 to 7%. And on average, you're getting about a percent SD change in your phenotype for an SD change in exposure. So if you come to me with a new association, say, oh, Chirag, I have this new connection between my favorite environmental factor and my favorite phenotype, and it's giving me roughly 2% increase, percent change for a change in phenotype, I can say that's roughly in the range of what you're getting in the entire span of the database, and that is not as strong as other factors that exist in the database that you might want to follow up with your hard-earned NIH money. So here's the kind of a view of the map that we depict here. So you can kind of see patterns that emerge from the data set, like, for example, nutrients connected with measures of adiposity, as well as metabolic indicators, as well as indicators of kidney function. Other things like PCBs also connected with things like metabolic function, hydrocarbons connected with inflammation. And if you punch all of these systematically into a model, you get about an R squared of about 20%. So 20% of change in a phenotype connected with changes of exposure in these data. So again, 80% of phenotypic variation left to explain from the large morass of environmental factors that are not ascertained in the NHANES study. And so you can actually condition on some of these distributions. So for example, if you're interested in all adiposity measures, you can see how environmental factors stack up on those. So here's the distribution of the effect sizes for these adiposity measures for different environmental exposures. So for example, you could see if your factor has about a 5% increase change in one of your phenotypes, how that stacks up with others. And you can do that with the other axes as well. So again, I think that one of the primary challenges is we need to be explicit in the number of hypotheses we test in our large databases. That is, reporting the false discovery rate. The family-wise error rate is another way of mitigating type 1 error. But at the very least, reporting the database size to our consumers. That is to say, how many things that we actually measured and what is our expected signal-to-noise threshold. But I think this new era of exposome research will give us new ways of understanding if your correlation matters or not, and how your new correlation that it might emerge compares to the entire list of correlations that you've derived. And here I give an example. For example, if your saturated fatty acids and BMI effects are affected only by 0.5%, does it really matter if 1.2% is on average in your database? So again, in summary, you will be able to find elusive environmental factors in phenotype and discover the elusive phenotypic variation in the population. Got to consider this as a totality of evidence and ask the question if your correlation matters in your database. But I also think that in genomic science today, there's a strong culture of reproducible research and this data literacy. And I think if you're interested in these topics, we could definitely talk. We've attempted to address some of these issues by putting all our code and asking individuals to try and reproduce our results. So here, Nam has put together a Docker container, our code that we use to produce some of these results from one of the papers that we show, and I think this is one way of kind of learning about how to do this type of research and improving on it. So I think my recommendations for designing a children's study are the following based on some of these genomic and large-scale data science exercises that we have done with genomics and environmental science. And that is, I think we need to increase sample sizes and make the data publicly available and measure genetics to discover the role of environment and phenotype. So increasing sample size, I think this these are great examples of how we've increased sample sizes and made the data publicly available through efforts like the UK Biobank and now emerging the China Kadoorie Biobank of sample sizes of almost in combination millions or so participants. And these, what they've been able to do is generate wide interest and visibility in this type of research. So already the UK Biobank, they've just released their 500,000 cohort, and already on bioRxiv, our preprint server, we've seen papers appear about using these data already. And so these are going to be hot data sets to do data science. And also it'll enable us, by having these data sets in the public domain, enhance reproducibility and potentially decrease false positives. So I think also measuring genetics is important to discover the role of exposures in phenotype. So there's several reviews that I've been inspired by that I don't have time to talk about, or include some work here reviewed by Greg Gibson, looking at how environments contributes to changes in gene function, how we can look at gene-environment interaction to tease out mechanistic changes that we've recently reviewed, as well as looking at new ways of doing GWAS to tell us about exposures, such as doing what is called a Mendelian randomization to find instrumental variables connected with our exposures to then connect to phenotypes in a causal fashion, kind of like how economists do instrumental variable analysis. So in conclusion here, I think that data sciences approaches that have been used in the genomic sciences will enable us to discover elusive phenotypic variation. I showed you things of how we could do it in mortality and some quantitative traits. But one of the issues are, of course, the dependencies of time and exogenous factors that induce this dense correlational web. And so we have issues such as these dense correlations, confounding, of course, an issue that I didn't really talk about, reverse causality, and how to assess this at high dimension will become the new reason for the season for data scientists. But I do believe by assessing things comprehensively will enable us to mitigate the fragmented literature of associations that John Schoenfeld and Ioannidis talk about. And I think that measuring genetics alongside exposures will probably prove useful for understanding causation. So in conclusion, I thank the folks who have helped me with this work, mostly Jake and Raj and Nam in the purpose of this presentation. And I also, again, thank the symposium organizers, the Ministry of Environment, and my funding sources for supporting my work. Thanks a lot.